Myself, I came into this field when I was a biologist doing research in marine biology, and I came across marine algae which was not producing spores, which you can easily see, but instead of producing spores, producing much smaller structures, mini spores. At this time, when I saw the pictures in the electron microscope, I saw thousands, millions of those particles, and they look like we are thinking viruses do look like. And so I believed that I have discovered, isolated, a harmless virus. Because the algae producing these particles, here in this picture you will see an enlargement on the right side. On this next photograph you will see the isolated particles. I thought I'm the first to have discovered and established a stable virus-host relationship. So I became virologist. And even so, having something in my hand that was not doing harm, but is in the end a useful structure. On the other hand, I believed in dangerous viruses and were teaching students to take care in front of HIV and better using double condoms than nothing, no words. In the end, I learned to characterize such a structure, isolating it, making photographs inside the organism, and isolate the particles outside the organism, and did biochemical analysis of its components, the proteins and the nucleic acid. And quickly I was informed by an Austrian professor that something is wrong with HIV and AIDS, because he asked me if I did check the evidence, and I told him no, the whole world is saying that HIV does exist and it's dangerous. And he said he was not asking the world. And I asked him, why you are adopting this idea? And he said he heard some rumors that something definitely it's wrong in this field. So he asked me to go into the library and to check the evidence on HIV, which at this time had another name, on my own. And so I came into this field, and I quickly realized, step by step, that in order to isolate a particle and to have it in the hand, that they went into electron microscopy, and they are presenting us photographs from dots of tissue, where everybody sees the big part of the dot of the tissue, and smaller particles, claiming those particles do represent viruses. But no control experiments. If those particles would be spherical and not belongings of the tissue dots, like documented here, then it should be seen that when you do a series of electron microscope around the sphere, that you'll see nothing, and then you see a smaller circle, a bigger circle, then a smaller circle again, and then nothing. Whereas in all photographs you always see a part of a tissue and only particles which are spherical. And this, it was very clear to me, where I isolated a structure here and thought that they are present in the organism, in the marine algae. And I really quickly found that none of the electron microscopic photos representing viruses are inside organisms. There is no single photograph on Earth saying that this particle has been found in a human being, in an animal, or in a liquid of this person. And of course, there is no presentation that they are isolated in the meaning of the word isolation, and no biochemical characterization of its molecules. And as I showed you, I spent a lot of time on the electron microscope, so I know all those techniques, and I quickly realized that they are showing us things which were not isolated, but were typical part of tissues dying in the test tube. And from those photos, they make a model, for example here the measles virus, and claiming this represents a virus. Whereas they never isolated something and they never showed the whole piece. And to get even worse, what they are doing, since the genetic area, since it is possible to multiply nucleic acid, to sequence nucleic acid, what they are doing, they take small parts of nucleic acid out of dying tissues, they sequence them, they determine the composition, and just mentally using software programs, they make a strand out of single small pieces, and the product at the end, they are naming a viral genome. And this is easy like this. And it's easy to show that it's wrong, and that's a misconception. And this is a self-fooling, and the virologists are fooling themselves, and they are fooling the public. It's not on bad intention, it's just because they are completely anti-scientific. They never ever tried to question themselves, and they never ever followed the first rule in science to perform and to document control experiments in order to show and to exclude the possibility that the results are the product of the techniques what you are using. So I'm able to state here right and now that all virologists claiming viruses which should cause disease are anti-scientific. Therefore, the laws applying corona means or the duty to get vaccinated are not justified, because they all are done under presumption that there is a scientific work out there. Therefore, all the laws on infectious diseases are violated by the fact that there is no science, but there is pure, easy to see, easy visible anti-scientific behavior and self-fooling.